How populations evolve. The number of known species has increased greatly over time. A scholar who sought to describe all the types of plants known to the Greeks in 300 BC had only about 500 different species to distinguish. In contrast, today, scientists recognize roughly 400,000 different plant species. The scientific community eventually agreed on a scheme introduced by Carolus Linnaeus, a Swedish scientist. His system, which is still in use today, is the basis for taxonomy. Taxonomy is the branch of biology concerned with identifying, naming, and classifying species. In the Linnaean system, each species is given a two-part Latinized name, or binomial. The first part is the genus, which is a group of closely related species. The second part of a binomial is used to distinguish the species within the genus. The two parts must be used together to name a species. Linnaeus also introduced a system for grouping species into a hierarchy of categories. The first step of this classification is built into the binomial. Then it places similar genera in the same family, puts families into orders, orders into classes, classes into phyla, phyla into kingdoms, and finally kingdoms into domains. The Greek philosopher Aristotle held the view that species were relatively fixed, or permanent forms that do not change over time. In the 1600s, religious scholars used biblical accounts and estimated the age of the Earth to be only 6,000 years old. Thus, the idea that all living species came into being relatively recently and are unchanging in form dominated the intellectual climate of the Western world for centuries. The study of fossils began to unravel the mystery behind the evolution of species. Fossils are imprints or remains of organisms that lived in the past. Stunning discoveries in the early 1800s included fossilized skeletons of a gigantic sea creature dubbed the ichthyosaur or fish lizard. This discovery convinced many naturalists that extinctions had indeed occurred. Ichthyosaurs were marine reptiles that were more than 50 feet long, and they ruled the oceans for about 155 million years before becoming extinct about 90 million years ago. One key feature of the ichthyosaur is its enormous eye, which is thought to be an adaptation to the dim light of the deep ocean. Naturalists compared fossil forms with living species and noted patterns of similarities and differences. In the early 1800s, French naturalist Jean-Baptiste Lamarck suggested that the best explanation for these observations is that life evolves. Lamarck explained evolution as the refinement of traits that equip organisms to perform successfully in their environments. He proposed that by using or not using its body parts, an individual would develop certain traits and then pass that on to offspring. Although Lamarck's idea of how species evolved was mistaken, his proposal that species indeed evolve as a result of interactions between organisms and their environment helped to set the stage for Darwin. Charles Darwin published a book called On the Origin of Species by Means of Natural Selection in 1859. The origin of species is a theory proposed by Darwin to explain the origin of species and to describe and organize the diversity of life. At the age of only 22, Charles Darwin began a sea voyage on the HMS Beagle that helped him frame his theory of evolution. The Beagle was a survey ship, and its main task was charting poorly known stretches of the South American coast. Darwin collected thousands of specimens of fossils and living plants and animals. He kept detailed journals of all of his observations. Many of Darwin's observations indicated that geographic proximity is a better predictor of relationships among organisms than the similarity of an environment. For example, the plants and animals living in temperate regions of South America more closely resembled species living in tropical regions of that continent than species living in similarly temperate regions of Europe. Darwin spent a lot of time at the Galapagos Islands. The Galapagos are relatively young volcanic islands about 900 kilometers or 540 miles off the Pacific coast of South America. Most of the animals that inhabit these remote islands are found nowhere else in the world, but they resemble South American species. Darwin noticed that Galapagos marine iguanas, with a flattened tail that aids in swimming, are similar to, but distinct from, land-dwelling iguanas on the islands and on the South American mainland. Each island had its own distinct variety of giant tortoise. Over time, differences gradually accumulated by the process that Darwin called descent with modification, which was his phrase to describe evolution. Darwin proposed scientific mechanism for how life evolves is natural selection. In natural selection, individuals with certain inherited traits are more likely to survive and reproduce than are other individuals with other traits. Darwin hypothesized that as a species spreads into various habitats over millions of years, natural selection results in diverse modifications, or evolutionary adaptations, that fit them to specific ways of life in their environment. Darwin published The Origin of Species, which is a book that supports his hypothesis with immaculate logic and hundreds of pages of evidence drawn from observations and experiments in biology, geology, and paleontology. Fossils are imprints or remains of organisms that lived in the past. They document differences between past and present organisms and show that many species have indeed become extinct. The soft parts of a dead organism usually decay rapidly, but the hard parts of an animal that are rich in minerals such as bones, teeth, and the shells of clams and snails may remain as fossils. Some fossils are called casts. A cast forms when a dead organism that was buried in sediment decomposes and leaves an empty mold that is later filled by minerals dissolved in water. The minerals then harden within the mold, making a replica of the organism. In rare instances, an entire organism, including its soft parts, is encased in a medium that prevents decomposition. Examples include insects trapped in amber and mammoths frozen in ice or preserved in bogs. Many fossils are found in fine-grained sedimentary rocks formed from the sand or mud that settles to the bottom of lakes, seas, swamps, and other aquatic habitats covering dead organisms. Over millions of years, new layers of sediment are deposited atop older ones, and it compresses them into a layer of rock called strata. Because younger strata are on top of older layers, the relative ages of fossils can be determined by the layer in which they are found. As a result, the sequence in which fossils appear within layers of sedimentary rock is a historical record of life on Earth. However, there are some limitations of the fossil record. For example, many of Earth's organisms did not live in areas that favored fossilization. Many fossils that did form were in rocks that were later distorted or destroyed by geologic processes. And not all fossils that have been preserved are accessible to paleontologists. Similarity resulting from common ancestry is known as homology. Homology analyzes similarities among different organisms. For example, when comparing the forelimb of a human, a bat, a whale, and a cat, the function of these forelimbs differ widely. A whale's flipper does not do the same job as a bat's wing. So if these structures had been uniquely engineered, we would expect that their basic design would be different from one another. The logical explanation instead is that the arms, forelegs, flippers, and wings of these different mammals are simply variations on an anatomical structure of an ancestral organism that over millions of years has become adapted to different functions. Biologists call such anatomical similarities in different organisms homologous structures. Homologous structures are features that often have different functions but are structurally similar because of a common ancestry. The hypothesis that all mammals descended from a common ancestor predicts that their forelimbs, though diversely adapted, would be variations on a common anatomical theme. The evolutionary history of each species is documented in the DNA inherited from its ancestral species in a continuous flow of genetic information across time. If two species have homologous genes with sequences that closely match, biologists conclude that these sequences must have been inherited from a relatively recent common ancestor. Conversely, the greater number of sequence differences between species, the more distance their last common ancestor is thought to be. Darwin believed that all life forms are related to one another. It is likely that all species descended from a common ancestor that used the DNA code. Organisms as dissimilar as humans and bacteria share homologous genes inherited from a very distant common ancestor. Geneticists have also uncovered hidden molecular homologies. Organisms may retain genes that have lost their function through mutations, even though homologous genes in related species remain fully functional. Vestigial structures are leftover structures that are of marginal or perhaps no important use to the organism. Vestigial structures are remnants of features that once served important functions in the organism's ancestors. For example, the small pelvis and hind leg bones of ancient whales are vestiges or traces of their walking ancestors. Another example are the eye remnants that are buried under scales in blind species of cavefish. Goosebumps are thought to be vestigial structures in humans. When humans become cold or agitated, we often get goosebumps caused by small muscles under the skin that make the body hair stand on end. The same response is visible and more functional in a bird that fluffs up its feathers for insulation. Comparative embryology also supports evolution. At some point in their development, all vertebrate embryos have a tail and pharyngeal pouches. These pouches are homologous structures that ultimately develop to have very different functions, such as gills in fishes and parts of the ears and throat in humans. Darwin was the first to visualize the history of life as a tree. The common trunk represents the first organism. The tips of millions of twigs represent the species living today. At each fork of the evolutionary tree is an ancestor common to all evolutionary branches extending from that fork. For example, let's look at an evolutionary tree of tetrapods, which are four-limbed animals. This scheme includes amphibians, mammals, and reptiles, including birds, and their closest living relatives, the lungfishes. In this diagram, each branch point represents the common ancestor of all species that descended from it. For example, lungfishes and all tetrapods are descended from an ancestor in step 1. 3 homologies are marked by the blue dots on the tree: tetrapod limbs, the amnion, which is a protective embryonic membrane, and feathers. Tetrapod limbs were present in the common ancestor in step 2, and hence are found in its descendants, the tetrapods. The amnion was present in ancestor at step 3, and thus is shared only by animals and reptiles, which are known as amniotes. Feathers were present only in ancestor step 6, and hence are only found in birds. Darwin knew that he would not be able to study the evolution of a new species by direct observation, but he did have a way to gain insight into the process of incremental change by observing the practices used by plant and animal breeders. Artificial selection is the selection of breeding domestic animals and plants to promote the occurrence of desirable traits in the offspring. All domesticated plants and animals are the products of selective breeding or artificial breeding from wild ancestors. Artificial selection has 2 essential components: variation and heritability. Variation among individuals, for example differences in coat type in a litter of puppies, allows the breeder to select the animals with the most desirable combination of characteristics as breeding stock for the next generation. Heritability refers to the transmission of a trait from parent to offspring. If a character is not heritable, it cannot be improved by selective breeding. Darwin found inspiration in an essay written by economist Thomas Malthus, who contended that much of human suffering, including disease, famine, and war, was the consequence of human populations increasing faster than food supplies and other resources. The production of more individuals than the environment can support leads to a struggle for existence, with only some offspring surviving in each generation. The essence of natural selection is the idea of unequal reproduction. It is the unequal survival and reproduction that results from the presence or absence of particular traits. In the process of natural selection, individuals whose traits better enable them to obtain food, escape predators, or tolerate physical conditions will survive and reproduce more successfully, and thus pass the adaptive traits to their offspring. Darwin reasoned that if artificial selection can bring about significant change in a relatively short period of time, then natural selection could modify species considerably over hundreds or even thousands of generations. Over vast spans of time, many traits that adapt a population to its environment will accumulate. If the environment changes, however, or if individuals move to a new environment, natural selection will then select for new adaptations to the new conditions, sometimes producing changes that result in the origin of a completely new species. The evolution of pesticide resistance in mosquitoes is an unsettling example of natural selection in action. Pesticides control insects and prevent them from transmitting diseases or eating crops, but pesticide resistance has evolved in hundreds of insect species. Whenever a new type of pesticide is used to control pests, the outcome is similar. Let's look at how this happens. Mosquitoes are first sprayed with a pesticide. A relatively small amount of poison initially kills most of the insects in the population. However, a few individuals carry an alternative form of a gene or allele that enables them to survive such a chemical attack. These genetically resistant survivors then reproduce, passing the allele for the pesticide resistance to their offspring. Thus, subsequent applications of the same pesticide are less and less effective, as the proportion of pesticide-resistant individuals increases in every generation. One thing to keep in mind, individuals themselves do not evolve. It is the population or the group of organisms that evolves over time as adaptive traits become more common in the group and other traits change or disappear altogether. Evolution refers to the generation-to-generation changes in populations. A population is a group of individuals of the same species that live in the same area and interbreed. In studying evolution at the population level, biologists focus on the gene pool. The gene pool consists of all copies of every type of allele at every locus in all members of that population. Imagine a wildflower population with two varieties of blooms that are different colors. An allele for red flowers, which we will symbolize by capital R, is dominant to an allele for white flowers, symbolized by lowercase r. Let's say that 80% or 0.8 of all flower color loci in the gene pool have the capital R allele. We'll use the letter P to represent relative frequency of the R allele in the population. So P equals 0.8. Because there are only two alleles in this example, the lowercase r allele must be present at the other 20% or 0.2 of the gene pool's flower color loci. Let's use the letter Q for the frequency of the lowercase r allele in the population. For the wildflower population, it would be Q equals 0.2. Because there are only 2 alleles for flower color, we can express the frequencies as follows. P plus Q equals 1. From the frequencies of alleles, we can also calculate the frequencies of different genotypes in the population if the gene pool is completely stable or not evolving. Now we can write a general formula for calculating the frequencies of genotypes in the gene pool from the frequencies of alleles and vice versa. This formula is called the Hardy-Weinberg equation, named for the 2 scientists who derived it. This is p squared plus 2pq plus q squared equals 1, where p is the frequency of homozygotes for one allele, pq is the frequency of heterozygotes, and q is the frequency of homozygotes for the alternate allele. Consider phenylketonuria, or PKU. PKU is an inherited inability to break down the amino acid phenylalanine. If untreated, this disorder has serious effects on the brain development. Newborn babies are now routinely tested for PKU, and symptoms can be prevented if individuals living with the disease follow a strict diet limiting phenylalanine. PKU occurs in about 1 out of every 10,000 babies born in the US. So, for 1 PKU occurrence per 10,000 births, our Q squared value is 0.0001. PKU is caused by a recessive allele, that is, one that must be present in 2 copies to produce the phenotype. So Q is the frequency of the recessive allele in the population, which equals the square root of 0.0001 or 0.01. P is the frequency of the dominant allele, which equals 1 minus Q, or 0.99. Small changes in the gene pool can be called microevolution. Evolution can be measured as changes in the genetic composition of a population over time. A non-evolving population is in genetic equilibrium, which is also known as Hardy-Weinberg equilibrium. The population's gene pool remains constant. There are 3 main causes of evolutionary change. These are natural selection, genetic drift, and gene flow. Natural selection is the only process that promotes adaptation. Populations consist of varied individuals, and some variants leave more offspring than others. For example, rabbits might prefer to eat red flowers over white ones. As a result, plants that produce white flowers will survive better and produce more offspring. One requirement for a gene pool to maintain the status quo is to have a large population size. The gene pool of a small population may not be accurately represented in the next generation because of sampling error. This evolutionary mechanism, a change in the gene pool of a population due to chance alone, is called genetic drift. What would cause a population to shrink down to a size where genetic drift is possible? There are 2 ways that genetic drift can come about. The first is the bottleneck effect, and the second is the founder effect. Disasters such as earthquakes, floods, and fires may kill large numbers of individuals. This produces a small surviving population that is unlikely to have the same genetic makeup as the original population. By chance, certain alleles may be overrepresented among survivors. Other alleles may be eliminated or underrepresented. Chance may continue to change the gene pool for generations until the population is again large enough for sampling errors to be insignificant. The founder effect occurs when a few individuals colonize and isolate an island, lake, or other new habitat, and the genetic makeup of the colony is only a sample of the gene pool in the larger population. The smaller the colony, the less likely it is to be representative of all genetic diversity that was present in the original population. If a colony succeeds, genetic drift will continue to change the frequency of alleles randomly until the population is long enough for genetic drift to be minimal. The type of genetic drift resulting from the establishment of a small new population whose gene pool differs from that of the parent population is called the founder effect. Gene flow is genetic exchange with another population. A population may gain or lose alleles when fertile individuals move into or out of the population. For example, suppose a neighboring population consists of entirely white-flowered individuals. A windstorm may blow pollen to our wildflowers from neighboring populations, resulting in a higher frequency of the white flower allele in the next generation. This would be an example of a microevolutionary change. Gene flow tends to reduce differences among populations. If it is extensive enough, gene flow can eventually join neighboring populations into a single population with a common gene pool. As people began to move about the world more freely, gene flow became an important agent of microevolutionary change in human populations that were previously isolated. Sexual selection is a form of natural selection in which individuals with certain traits are more likely than other individuals to obtain mates. Because sexual selection has a direct impact on relative fitness, it is an especially powerful form of natural selection. Males and females of an animal species have different reproductive organs, but they may also have secondary sexual traits that are noticeable differences not directly associated with reproduction or survival. The distinction in appearance, called sexual dimorphism, is often manifested in size difference. Among male vertebrates, sexual dimorphism may be evident in adornment, such as manes on lions, antlers on ears, and colorful plumage on peacocks and other birds. If natural selection favors certain bird color phenotypes over others, then the population of mice will change over the generations. 3 general outcomes become possible, depending on which phenotypes are favored. These modes of natural selection are directional selection, disruptive selection, and stabilizing selection. Directional selection shifts the overall makeup of a population by selecting in favor of one extreme phenotype, for example, the darkest mouse, or the most colorful and largest plumage. Directional selection is common when the local environment changes, or when organisms migrate to a new environment. An actual example of directional selection is the shift of insect populations towards a greater frequency of pesticide-resistant individuals. Disruptive selection can lead to a balance between 2 or more contrasting phenotypes in a population. A patchy environment, which favors different phenotypes in different patches, is one situation associated with disruptive selection. Stabilizing selection favors intermediate phenotypes. Such selection typically occurs in relatively stable environments, where conditions tend to reduce physical variation. This evolutionary conservatism works by selecting against the more extreme phenotypes. Of the 3 selection modes, stabilizing selection occurs in most situations. For example, stabilizing selection keeps the majority of human birth weights between 3 and 4 kilograms. For babies much lighter or heavier, infant mortality is greater. Thank you for watching.